Hello students, today I bring for you a new chapter and an important one, that is Principles of Inheritance and Variation. In this chapter, we are going to cover topics like Mendel's law of inheritance, inheritance of one gene, inheritance of two genes, sex determination, mutation and genetic disorder. We will cover NCERT as well as this PDF. So let's start this chapter. First we are going to see about what is the topic inside the PDF. First of all, Mendel's law of inheritance. You have to start with this some important terms, and that are what is genetics. Genetics means study of inheritance, heredity and variations of characters, or study of genes or chromosomes is called genetics. Coming to inheritance, it is the transmission of characters from parents to progeny. It is the basis of heredity. Progeny means offspring. Coming to variation, that is difference between parents and offspring. Coming to character, it is the heritable feature among the parents and offspring. Example, eye color. And finally, traits. Traits are the variants of a character, that are brown eye, blue eyes, etc. Next, alleles or allelomorphs. It is the alternative form of a gene. Example, capital T for tallness and small t for dwarfness are two alleles of a gene for the character height. Coming to homozygous, it is the condition in which chromosomes pair carry similar alleles or a gene of a gene, also known as pure line, that is true breeding. Example, capital T, small t, capital Y and small y, etc. And then coming to heterozygous, it is the condition in which chromosome pair carries dissimilar alleles of a gene. Example, capital T, capital Y, etc. Here you can see in this diagram, a chromosome pair is given as you can see. It is a homozygous one. It is the homozygous one, heterozygous as you can see, and homozygous. Capital B and small b are two alleles of a character. Next is dominant character. Here the character which is expressed in heterozygous condition, it is indicates with a capital letter, that is dominant one. Coming to recessive character, it is the character which is suppressed in heterozygous condition, it indicates with the small letter. Phenotype is the physical expression of a character. Genotype is the genetic constitution of a character, and hybrid is an individual produced by mating of genetically unlike parents is called hybrid. Here you can see some examples are given: dominant character and recessive character. The clipped chin and no clipped, as you can see differences. Widow's peak, no widow's peak. Here you can see it is a widow's peak, and here no widow's peak is there. Dimples, no dimples. Brown or black hair, blonde hair. Freckles, no freckles. Brown eyes, grey or blue eyes. Free earlobe and attached earlobe. These are the some dominant recessive characters found in a human being. Let's start. Gregor Mendel, father of genetics. And coming to Mendel's law of inheritance, Mendel conducted some hybridization experiments on garden pea, that is Pisum sativum, for 7 years, 1856 to 1863. And steps in making a cross in a pea. As you all know that Mendel selected the garden pea plant for his experiments. So why he selected and how the process he take to proceed this experiments. First of all, selection of two pea plants with contrasting characters. First, he have to select pea plants with contrasting characters. Second is emasculation, that is removal of anther of one plant to avoid self-pollination. As you can see, this process is emasculation. This is female parent. Coming to pollination, collection of pollen grain from the male parent and transferring to the female parent. And then finally collection and germination of seed to produce offspring. These are the steps in making a cross in a pea plant. Next, Mendel chose pea plant because pea plant have 7 true breeding varieties of contrasting characters or traits. As you all know, characters also called traits. First of all, stem height. Height of the stem. As you can see, tall and dwarf, where the tall is dominant and dwarf is recessive. Flower position, axial and terminal. Here you can see flower position, axial means sidewise, terminal means in top position. Flower color, violet or white, like this. Pod shape, inflated, constricted. Inflated means like a flattened pod, as you can see, and constricted like this. Okay. Pod color, green or yellow. Here. Seed shape, round, wrinkled. This one. And seed color, yellow and green, like this one. That's why Mendel selected pea plant for his experiment, because it shows a proper contrasting character, visible contrasting character. Next, inheritance of one gene, that is monohybrid cross. A cross involving two plants differing in one character pair. In one character pair, that's why it is called monohybrid cross. Example, Mendel crossed tall and dwarf pea plant to study the inheritance of one gene. Tall and dwarf. Here you can see, there is a cross between tall and dwarf plant, cross between tall and plant and dwarf plant. And here seeds are also there, cross between yellow seeded plant or green seeded plant. Only one character Mendel taken. According to your NCERT, the monohybrid cross belongs to only one character, contrasting character, that is a tall and dwarf. Here, parent plant is homozygous tall, that's why capital T mentioned, that is pure breed, and homozygous dwarf, that is pure shortness, small t and small t. These are the gametes he crossed in the first filial generation. First result is coming to like this, capital T and small t. It is the plant showing tallness, because capital T for tallness and T is here, that is dominant, small t is recessive. So the homozygous dwarf parent's character, that is hidden in the first generation. That's why it is called heterozygous tall, impure tall. Then Mendel again F1 plant selfing, heterozygous tall, as you can see, heterozygous tall, again cross between them, gametes are like this. And in F2 generation, second filial generation, what is the result came? Here, one pure tall plant, two impure tall plant, and one dwarf plant. This type of square is called punit square or checker board also. Here what is the result came? As you can see, in monohybrid phenotypic ratio will be tall will be 1 and dwarfness will be, tall will be 3 and dwarfness will be 1, 3:1. Phenotyping means the characters present while you are seeing a plant and how you can identify the differences between them. That is called phenotyping. From externally observing character is called phenotyping. So Mendel found out tallness will be 3 number and dwarfness will be 1, 3:1 came, as you can see from this punit square. And coming to genotypic ratio, homozygous tall will be 1, heterozygous tall will be 2 and homozygous dwarf will be 1. 1:2:1 is the genotypic ratio. Genotypic means there is a modification inside the characters of the plant, that is called genotypic. And phenotypic means the observable character by watching or seeing a plant, that is called phenotypic ratio, phenotypic character. Here you can see inheritance of one gene. Mendel made similar observations for the other pair of traits. Not only the tallness or darkness, Mendel also observed other pair of characters by taking other pair of characters like flower color, seed shape, pod shape, flower position, plant height. Plant height already taken. So he found out the same ratio, as you can see. In F2 generation, dominance will be like this number, receive from this number. The yellow seed will be this number, receive from this number. Seed shape, round, wrinkled, pod color, green color and yellow one. Here pod shape, round and constricted, flower position, axial and top, and plant height also mentioned here. So you can also identify by seeing this table that in everything there is a particular ratio, 3:1 ratio is there, as you can see. So he proposed that some factors are also called genes, now called genes. He proposed that some factors were inherited and from parents to the offspring, and factors are now called genes. So what Mendel concluded, you can see the F1 or the F1 result, capital T and small t, heterozygous tall, when self-pollinated, produce two gametes, capital T small t in equal proportion. And during fertilization, pollen grains of capital T have 50% chance to pollinate egg of the capital T and small t. Also pollen grains of genotype small t have 50% chance to pollinate eggs of capital T and small t. So here you can see 1/4 of the random fertilization leads to capital T, half of the random fertilization leads to capital T small t, and 1/4 of the random fertilization leads to small t and small t. That is 1/4 capital T, half small t, capital T small t, and 1/4 small t and small t. And the ratio will be 1:2:1. Here again, by using binomial expression ax plus by whole square, hence with this result you can see this result will become. And Mendel self-pollinated the F2 plant. He found that the dwarf F2 plant continued to generate dwarf plant in F3 and F4 generations. He concluded that the genotype of the dwarf plant was homozygous, that is small t and small t. Before starting the next topic, let's see what your NCERT says here. First of all, inheritance is the process by which characters are passed on from parents to progeny. It is the basis of heredity, and variation is the degree by which progeny differ from their parents. Here some things are given that will be not important. First of all, here you can see it was during the mid-19th century that the headway was made in the understanding of the inheritance. Gregor Mendel conducted hybridization experiments on garden pea for 7 years, that is 1856 to 1863, and proposed the law of inheritance in living organism. During Mendel's investigation into inheritance pattern, it was for the first time that statistical analysis and mathematical logic were applied to problem in biology. Before that, no one using this type of mathematical logics in biology. Mendel started for the first time. Then Mendel chose garden pea plant and manifested as two opposite traits, tall and dwarf plants. First of all, yellow and green seeds, and with this also Mendel have found out that the pea plant have 14 two breeding plant varieties. You already seen, and also here mentioned you can see, and the 14 traits are stem height, tall and dwarf, flower color violet white, flower position axial terminal, pod shape inflated constricted, pod color green yellow, seed shape round wrinkled, and seed color yellow and green. Inheritance of one gene. Here what happened? The first generation is called first hybridization, also called filial one progeny or F1 generation. Then here you can see the proportion of plants that were dwarf were 1/4 of the F2 plant, while 3/4 of the F2 plant are were tall. The tall and dwarf traits were identical to their parent type, and did not show any blending. Blending means mixing. This is all, that is all the offspring were either tall or dwarf. None of were in between the height. Remember, none of them were in between the height. And similarly, we obtained with the other traits Mendel studied, and same result also came, and the proportion will be 3:1. Next, here you can see Mendel found out that something is transferred from parents to offspring, and that something is given the name factors. Now we call them genes. So genes are the units of inheritance. They contain information that is required to express a particular trait or character in an organism. Genes which code for a pair of contrasting traits are known as alleles. That is, they are slightly different from the same gene. Here, same thing is given with proper explanation. As you can see, coming to alleles, capital and small t are alleles of each other. Hence, coming to pair of the height, it would be capital T, capital T small t, or small t, small t. Mendel also proposed in the true breeding plants, that is tall or dwarf variety, that allelic pair of genes for height will be identical, that is homozygous. If it is identical, that is homozygous, like this. And if it is non-identical, then that is called heterozygous. See, heterozygous also mentioned here, and recessive, dominant recessive also given. Then Mendel found out the monohybrid cross, and here is the diagram. As you can see, Mendel taken true breeding parent plant. It is the parent generation, and it is the first filial generation or F1 generation, and then F2 generation. And with this cross, here it is given, when the tall and dwarf plant produce gametes by the process of meiosis. Obviously simple, meiosis will occur to produce gametes. We already know about this. The alleles of the parental pair separate or segregate from each other, and only one allele is transmitted to a gamete. Here with this experiment, we also find out that the alleles of a parental pair separate or segregate from each other, and only one allele is transmitted to the gamete. And the segregation of alleles is random process, and so there is 50% chance of gametes containing either allele. And here you can see, if they are true one or pure one, then they have capital T or small t, that is pure tall and pure dwarf. And if they have impure, they will capital T and small t. In other words, hybrids they are called. So, if the hybrid containing allele which express contrasting trait, that is called heterozygous. And this all particular diagram. Here is the particular square is given. It is called Punnett square, because it was developed by British geneticist Reginald C. Punnett. Then what it is given here, you can see same things are given, 50% again, many times written. And if you come into symbols, for these symbols are used to this one, the +1, it results the female or eggs, and this symbol, when arrow is sideways like this, is the male one. Next, same things are given, 3:1 ratio, genotypic ratio will be 1:2:1, and there will be binomial expression ax plus by square. We already seen in the PDF. And then some crosses are given, test cross, Beck cross, okay, which we will going to cover this. Next, shift it to the PDF. Here, coming to this PDF, as you can see, second part, that is Beck cross, test cross. What is Beck cross? Here it is given, cross between hybrid and its any parent. Here you can see hybrid one, that is the heterozygous tall, and a recessive parent or any parent plant we take in the recessive parent, and coming to the cross, here ratio will be 1:1. Coming to the test cross, that is crossing of an organism with dominant phenotype to a recessive individual. Crossing of an organism with dominant phenotype to a recessive individual. It is a test cross, as you can see. It is used to find out the unknown genotype of a character. It is a homozygous recessive. It is also homozygous recessive. As you can see, small ww is mentioned here, and here the genotype is unknown. As you can see, we have only dominant phenotype. So here the changement is given. Capital WW is taken, whereas in this type capital W and the small w is taken. And then by using a Punnett square, what is the result? In the first type, result are all flowers are violet. All flowers are violet. And here you can see in this type, half of the flower are violet and half of the flower are white. It is the result. Interpretation, unknown flower is homozygous dominant. So the flower, unknown flower which we have taken, it is the homozygous dominant, while the unknown flower in this type, it is heterozygous. Mendel conducted test cross to determine the F2 genotype. That is the test cross. Here. Finally, the first law of dominance. Mendel stated that characters are controlled by discrete units that are called factors. That it is a factor, this one also a factor, and factors occur in pairs. Factors always occur in pair. Here you can see similar pair of factor, here similar pair of factor, and dissimilar pair of factors, where one is dominant, one is recessive. If in a dissimilar pair of factor, one member of the pair will be dominant, that is dominant, the other, which is the recessive one. And coming to second law of segregation, here what Mendel says, that during gamete formation, the factors or alleles of a character pair present in parents segregate from each other such that a gamete receives only one of the two factors. As you can see, during gamete formation, or we already seen in meiosis topic, the factor or the genes of a character pair present in parents segregate or separate from each other such that a gamete receives only one of the two factors. Only one of the two factors. If it is homozygous parents produce similar gametes. Heterozygous parents produce two kinds of gametes, each having one allele and with equal proportion. Here you can see similar pair of factors, homozygous, similar gametes are formed. Similar pair of factors, homozygous, again similar gametes are formed. Dissimilar pair of factors, heterozygous, dissimilar gametes are produced. It is the second law or law of segregation. Next is dihybrid cross. We already seen about the monohybrid cross. What is the phenotypic ratio, genotypic ratio? We already completed. Here, dihybrid cross. It is a cross between two parents differing in two pair of contrasting character. Two pairs are taken. That's it is called dihybrid cross. In monohybrid cross, a single character is taken. Example, cross between pea plant with round shaped and yellow colored seed RR and YY. Round shape, dominant. Yellow color also dominant. That's why all are in capital letter. Wrinkled shaped or green colored seed, small RR and small YY, where both are the recessive one. Here you can see round yellow, this type of seeds, and wrinkled green, this type of seed. It is the dominant one, pure dominant. It is the pure recessive one. First of all, in dihybrid cross, parents we already selected homozygous round yellow, homozygous wrinkled green, and gametes are like by crossing this type of gametes are produced, and in F1 generation this is came capital R, small r, capital Y and small y, heterozygous round yellow. By selfing these two, what we find out, gametes will be like this: capital R, capital Y, capital R, small y, small r, capital Y, small r, small y, and this same also in this side. These are the gametes formed. Next, coming to F2 generation, again we use the Punnett square, and the results you can see. This will be crossed, and this is the result. Here again, this one cross with this one, result will be like this, and we have the full table, you can see. Then, on observing the F2 generation, Mendel found that the yellow and green colors segregate in 3:1 ratio. Round and wrinkled seed shape also segregate into 3:1 ratio, and here you can see 9 round yellow, 3 round green, 3 wrinkled yellow, 1 wrinkled green is present, and the phenotypic ratio will be 9:3:1 in dihybrid cross. This ratio can be derived as a combination of series of 3 yellow is to 1 green with 3 round is to 1 wrinkled. That is 3:1, 3:1, and the result will be 9:3:1. It is the phenotypic ratio of dihybrid cross. Again, if we find out the genotypic ratio, then what it will be? Here you can see this number will be 1, and this type of structure we can find out 2 are there, this one and this one, that's why it is mentioned 2. This structure also 2 are there, this one and this one. This structure 4 are there, like this, all 4 are here. So coming to the genotypic ratio of the dihybrid cross, though it is not mentioned in your NCERT, but it is important. The ratio will be 1:2:1:2, 1:2:4:2, 1:2:1:1. This is the dihybrid genotypic ratio. And then is the third law, that is law of independent assortment, where it is based on the result of dihybrid crosses. It states that when more than one pair of characters are involved in a cross, factor pair independently separate from the other pair of the characters. As you can see, segregation of factor pair, it is independently assorted. That is, you can see, find out the factor 1 pair, factor pair second, possibility 1, possibility 2. First of all, these two will make a pair, these two will make a pair like this, as you can see, and the result are here. It is the first possibility. And second one, these two will make a pair, these two also make a pair like this, as you can see, and the result are different from this one. So it is found out that when more than one pair of characters are involved in a cross, factor pairs are independently segregated from the other pair of the characters. Here, the concept of dominance. Every gene contains information to express a particular character or trait. In heterozygotes, there are 2 types of alleles. One is the unmodified, that is normal or functioning allele. It is generally dominant and represents the original phenotype, and a modified allele, it is generally recessive. As you can see, alleles of brown eye color is unmodified allele because it is found in many human beings. But alleles of blue eye color, it is a modified allele because it is found only in few. Here, unmodified allele and unmodified allele, they make the homozygous gene pair. Unmodified allele and a modified allele, they make the heterozygous gene pair, and modified allele, they make the homozygous gene pair. Here, on to the concept of dominance. Example, consider a gene that contains information for producing an enzyme. We all know that genes are responsible for production of the enzymes. Normal allele of a gene produce normal enzyme, and modified allele is responsible for production of three options are there: normal or less efficient gene enzyme, non-functional enzyme, or no enzymes at all. In the first case, if the modified allele produce normal or less efficient enzyme, in this case, modified allele will be produce the same phenotype like unmodified allele. The first condition, first result came. Second, thus modified allele is equivalent to unmodified alleles. Here, coming to the non-functional enzymes, in this case, the phenotype will be dependent only on the functioning of the unmodified allele. Thus, the modified allele becomes recessive. In case 2, you can see unmodified allele producing enzyme, modified allele producing non-functional enzyme. And while in case 3, unmodified allele produces enzyme, modified allele produces no enzyme. We will start in the part 3. Let's see what your NCERT says here. Same topics are given, you can see. And with this, based on the observation of monohybrid cross, Mendel proposed two general rules that are, these rules are called principles or law of inheritance. The first law will be law of dominance, second will be law of segregation. The law of dominance, characters are controlled by discrete units that are called factors, and factors always occur in pair. In a dissimilar pair of factor, one member of the pair dominate the other, that is recessive. And the law of dominance is used to explain the expression of only one of the parental character in a monohybrid cross in the F1 and the expression of the F2. It also explains the proportion of 3:1 to obtain at the F2 generation. And coming to law of segregation, we already seen in this diagram. And in the diagram, what happened? Again, revisit the PDF where the law of segregation. Here, during gamete formation, the factors of character pair present in parent segregate from each other such that a gamete receive only one of the two factors. That is the law of segregation. Then is incomplete dominance. Incomplete dominance. Before starting this, let's see what the PDF says. Here, all the topics are completed. Next is non-Mendelian inheritance, that is incomplete dominance, codominance, multiple allelism, polygenic inheritance, pleiotropy. First of all, incomplete dominance. Here, it is the inheritance in which the offspring show intermediate character between two parental characteristics. Means, parent's color will be different and the hybrid color will be different from both the parents. Example, flower color in snapdragon, dog flower or Anthrinum sp and Mirabilis jalapa, 4 o'clock plant. Here cross between homozygous red and white produces pink color flower, and pink does not mix with the red or a white one. It is called incomplete dominance. Remember the plant name: snapdragon or dog flower or Anthrinum, and Mirabilis jalapa, that is also called 4 o'clock plant. They show incomplete dominance. Here again detailed phenotype ratio 1:2:1, 1 red, 2 pink, 1 white. From this cross we can find out genotype ratio also 1:2:1, like this. Thus phenotypic and genotypic ratio are same. This means R, capital R, was not completely dominant over the small r. Pea plant also shown incomplete dominance in other traits. Next is codominance. Codominance means the inheritance in which both allele of a gene expressed in a hybrid. Both alleles are expressed. Example, ABO blood grouping in human being, where the first of all ABO blood group are controlled by a particular gene that is called I, capital I. It controls the production of sugar, polymer or antigens that protrude from plasma membrane of the RBC like this. Protrude means comes out like this. This one comes out. This one also, it is the RBC. And the gene I has three alleles: IA, capital I, capital A, capital I, capital B, and small I. Both IA and IB produce slightly different from the sugar. Allele small I does not produce any sugar. That's why in this diagram you can find out. Here blood type A produce antigen A, blood type AB produce antigen B, blood type AB produce antigen A and B, and blood type O produce neither antigen A nor B. Here you can see blood group genotype. In A blood group, genotype will be capital A, and again capital A like this, or this one, because small I will be the recessive one. In B also this one or this one. In AB blood group both are dominant, that's why it is called codominant. In O blood group only this one is the genotype. Here table also given in your NCERT. Allele from parent 1, first parent either father or mother. Here second parent, again father or mother. Genotype will be like this. If the father having this type of blood group, mother also this type, then the blood group will be this one. Blood type will be A blood type. Here the father having this type of blood group, mother having this type of blood group, then the result will be like this, and again A blood group will be produced in the offsprings. Same with the B blood group, same with AB blood group like this one. AB blood group is the codominance, because here both A and B blood group are codominants to each other. And in the last one, here what it will be? Allele like this, receive allele having the parent father, receive allele having the mother, then the result will be like this, and it will be O blood group. Here when I and IB are present together, they both express their own type of sugar. This is due to codominance. Codominance. Here again examples you can see. Father having this type of blood group, mother having this blood group. We crossed using the Punnett square, and these are the results. From this Punnett square, A blood group will be like this, B blood group, this one, AB blood group, this, and O blood group like this. All four are comes out. Here father having AB blood group, mother having O blood group, then this result will come. Offsprings will be either A blood group or B blood group. Next is multiple allelism. It means multiple allelism means the presence of more than two allele on a gene to govern the same character. More than two alleles of a gene to govern the same character. Example, alleles of ABO blood grouping, 3 alleles are there. In blood group AB, there are 3 alleles are present. In an individual, only 2 alleles are present. Multiple alleles can be found only in a population. Like this. Multiple like this. Fourth is polygenic inheritance. It means it is the inheritance in which some traits are controlled by several genes. Multiple genes also called. Example, human skin color, human height. It considers the influence of environments also. It is a polygenic trait. Phenotype reflects the contribution of each allele, that is the effect of each allele is additive. Like this. In a particular population, you can easily find out some skin color will be white, some have tint, hue, shade and black. Human skin color. Assume that three genes A, B, C control human skin color. You have to assume dominant form A, B and C responsible for dark skin color. Recessive form small a, B and C for light skin color. Then the genotype with all the dominant allele. It will be darkest skin color like this, darkest one. Genotype will be all the received alleles A, B, B and C, small one gives the lightest skin color, that is the more white, and genotype with three dominant, three recessive allele gives the intermediate skin color, this one, brown color. Thus, number of each type of allele in the genotype determines the darkness or lightness of the skin. Next is pleiotropy. Pleiotropy means here a single gene exhibit multiple phenotypic expressions. Such genes are called pleiotropic gene. In most cases, mechanism of pleiotropy is the effect of gene on metabolic pathway which contributes towards the different phenotypes. Example, starch synthesis in pea plant, phenylketonuria, sickle cell anemia, etc. Coming to example, in phenylketonuria and sickle cell anemia, the mutant gene has many phenotypic effects. Example, phenylketonuria causes mental retardation, reduction of hair and skin pigmentation. Like a single gene, pleiotropy, multiple traits having trait A, trait B and trait C. Here coming to the starch synthesis in pea plant, starch is synthesized effectively by this type of gene, capital B and capital B genes. Therefore, large starch grains are produced. While the small b and small b have lesser efficiency in starch synthesis and produce smaller starch grain. Starch grain size also shows incomplete dominance. Here you can see this one we taken effective starch synthesis, large sized seed and this small sized seed like this. Gametes are here as you can see and the result will be capital B and small b. Intermediate sized seed which is the incomplete dominance. Next, chromosomal theory of inheritance. Before starting this, let's go to the NCERT. Here you can see incomplete dominance. It is the same points. Dog flower is mentioned, snapdragon or anthrinum sp is a good example to understand incomplete dominance, where parent generation red colored flower, white colored flower, gametes are like this, but result will be all pink. Same things are mentioned here, nothing more. Here in incomplete dominance, both phenotypic and genotypic ratio are same. Next is codominance. Coming to codominance, you already seen in the blood group of human being, ABO blood grouping, where it is controlled by gene I. The plasma membrane of the red blood cell has sugar polymers that protrude from its surface, and this kind of sugar is controlled by the gene. The gene I has three alleles like this, and these two produce slightly different form of sugar, while allele small I does not produce any sugar. Because of humans are diploid organism, each person possesses any two of the three I gene alleles. I and IB are completely dominant over small I. And same examples are given. Here the table you can see. We already seen in the PDF. Here multiple alleles also in a ABO blood grouping we can easily find out the multiple alleles. Here occasionally a single gene produces more than one effect. Example, starch synthesis in pea plant is controlled by only one gene, and that will be B and B gene. And for large seed, this will be combination will be there, and for smaller seeds, smaller starch gain, this combination will be there, homozygous. And while maturation, This will be, seed will be round and this will be wrinkled. Next, coming to inheritance of two genes. Now, genes are inherited. Here the dihybrid cross is given. Same diagram is there. With proper shape you can find out. As I told you, there is no genotype ratio will be asked in exams, but that is for your extra knowledge. Coming to law of independent assortment, when two pair of traits are combined in a hybrid, segregation of one pair character is independent of the other pair of characters. That is called law of independent assortment. And Punnett square also very much helpful to easily find out. Next is chromosomal theory of inheritance. Let's see what the PDF says here. Why did Mendel's work remain unrecognized? First of all, communication was not easy at that time. Its mathematical approach was new and unacceptable by some biologists, and the concept of gene or factors as stable and discrete unit could not explain the continuous variation. Mendel said something is transferred from parents to offspring, but he did not practically explain that. And here he could not give any physical proof for the existence of factors. That's why his work will be unrecognized. And Mendel's words that my time will yet come, not now but afterwards. And see, in 90s, three scientists came, De Vries, Correns and von Tissermark independently rediscovered the Mendel's result. First of all, it is proposed by Walter Sutton and Theodore Boveri 1902, and they said that the pairing and separation of a pair of chromosomes leads to segregation of a pair of factors they carried. Sutton united chromosomal segregation with Mendelian principles and called the chromosomal theory of inheritance. So who gave the name chromosomal theory of inheritance? Now, Walter Sutton. Here you can see important statements. First of all, chromosomes are vehicles of heredity, and two identical chromosomes form a homologous pair. As you can see, homologous pair of chromosomes, paternal, maternal replication like this, sister chromatids like this. A homologous pair of chromosome, gene for eye color, gene for enzyme A. Two genes are there like this. Gene from cytochrome C, this one. From mother, from father. They both combined with each other. Like this here you can see. Homologous pair segregate during gamete formation. Homologous pair segregate or separate during the formation of gametes. As you can see, segregation of chromosomes like this. How they are segregated? See the color combination. It is also in your NCERT diagram as well as from here. And here also metaphase 1, metaphase 2 gametes are produced too. How they segregate with each other. Next, independent pairs segregate independently. Independent pairs segregate independently. Again some possibilities as you can see. Possibility 1, possibility 2, and here also it is the first possibility, it is the second one. Like this. With orange, it has the green color enzymes, and with yellow, they have the red color. But here it is different as you can see. And it shows the independent assortment. Genes are present on chromosomes, hence they show the similar behavior. And further, Thomas and Morgan worked with the fruit flies Drosophila melanogaster to prove the chromosomal theory of inheritance. Thomas Hunt Morgan. Remember, here you can see Thomas and Morgan worked with the fruit flies, that is Drosophila melanogaster, to prove prove the chromosomal theory of inheritance. And why he chose the Drosophila? Because they can grow on simple synthetic medium. Short generation time, life cycle will be 12 to 14 days. Breeding can be done throughout the year. Hundreds of progenies per mating, and male and female flies are easily distinguishable. Example, male is smaller than the female, like this. It has many types of hereditary variations that can be seen with low power microscope. Here the varieties: wild type, ebony, yellow, vestigial. That's why Morgan chose the Drosophila to prove the chromosomal theory of inheritance. Next is linkage and recombination. Linkage is the physical association of two or more genes on a chromosome. Physical association of two or more genes on a chromosome. Linked genes do not show independent assortment. Linked genes, they do not show independent assortment. Recombination is the generation of non-parental gene combination. Recombination occurs due to independent assortment of or crossing over. Here you can see the tightly linked genes and loosely linked genes. These are the tightly linked and these are the loosely linked genes. And here also diagram. Homologous chromosomes aligned like this. Chromosomal crossing over and recombinant chromatids. These are the recombinant chromatids. These are the non-recombinant chromatids. Here, Morgan carried out several hybrid crosses in Drosophila to study the sex-linked genes. Example, cross 1, yellow-bodied, white-eyed female. This one with the brown-bodied, red-eyed male, wild type, and this one. Another cross, white-eyed, miniature winged, this, and red-eyed, large winged, wild type, and this one. And the major findings of Morgan experiments are: the two genes did not segregate independently and the F2 ratio deviated from 9:3:3:1. Genes were located on X chromosome, and when two genes were situated on the same chromosome, proportion of parental gene combination was much higher than the non-parental type. This is due to linkage. What is the third point says? Important one. When two genes were situated on the same chromosome, proportion of parental gene combination was much higher than the non-parental type. This is due to linkage. See the diagram? It is the NCERT one. First cross, yellow and white, wild type, and during the cross you can see in F1 generation, gametes are parental character 98.7%, recombinant character 1.3% in F2 generation. And here in cross B, he chose the white miniature and wild type. Male and female also mentioned with diagram. In F1 generation, wild type white miniature, parental type 62.8%, recombinant type 37.2%. Next is genes of white eye and yellow body were very tightly linked and shown only 1.3% of recombination, while genes of white eye and miniature wing were loosely linked and shows 37.2% recombination. Tightly linked genes show low recombination and loosely linked genes show high recombination. Here also these are the tightly linked, as you can see, these are the loosely linked, and recombination also you can find out. Tightly linked genes, they show only 1.3% recombination, while the loosely packed genes show 37.2% recombination. And then Alfred Stewart band used the recombination frequency between the gene pair for measuring the distance between genes and mapped their position on the chromosome. Genetic map are used as a starting point in the sequence of genome. Example, Human Genome Project. And here you can see gene for body color, gene of eye color, gene of wing size, and the preserve and distance like this you can see. Total distance 17%, where gene of the body color and eye color will be 8% different distance, and the gene of the eye color to the wing size will be 9% differences distance. Sex determination. Before starting this, let's see what your NCERT says. Here, chromosomal theory. Again, the points are given. Mendel published his work of inheritance in 1865, but for several reasons it remained unrecognized till 1900. First of all, communication was not easy in those days, and his work could not be widely publicized. Second, his concept of gene or factor are stable and discrete units that are control the expression of traits. The pair of alleles which did not blend with each other was not accepted by his contemporaries as an explanation for the apparently continuous variations seen in nature. And thirdly, Mendel's approach using mathematics to explain biological phenomena was totally new and unacceptable to many of the biologists at that time. And finally, he could not provide any physical evidences that the factor and what they are made up of. But in 90s, three scientists DeVries, Correns and von Desarmark independently rediscovered Mendel's results on the inheritance of characters. And by that time, because of advance in microscopy, they also find out, easily identify cell division and how the chromosomes transfer, divisions are there, and chromosomes are named. By 1902, chromosome movement during meiosis has been worked out. Walter Sutton and Theodore Boveri noted that the behavior of chromosomes was parallel to the behavior of genes and used chromosome movement to explain the Mendel's law. And here again same diagrams is there. During anaphase of meiosis 1, the two chromosomes pair can align at the metaphase plate independently of each other. And here are the possibilities. One long orange and short green chromosome and long yellow and short red chromosomes at the same pole, as you can see. Here, one long orange and short red chromosome, long yellow and short green chromosome at the same pole. It is the same pole, okay. If I put a line in between like this, pole 1 and pole 2, as you can see. In meiosis 2 anaphase stage, what happened? Crossing over occur between them. They all are separated. Like this, the point mentioned here, it is not mentioned here. And germ cell is produced too. And with this, in past possibility, they align with the same pole or they crossing over occur. It is called independent assortment. Here, Sutton and Boveri argued that the pairing and separation of a pair of chromosomes would lead to the segregation of a pair of factors they carried. Then Sutton united the knowledge of the chromosomal segregation with Mendelian principle and called the chromosomal theory of inheritance. Then Thomas Hunt Morgan and his colleagues again do some experimental verification of chromosomal theory of inheritance by using the Drosophila melanogaster. And why he chose the Drosophila melanogaster? Because they could be grown on simple synthetic medium in a laboratory. They complete their life cycle in about 2 weeks. A single mating could produce a large number of progeny flies. Also, there will be a clear differentiation of the sexes. The male and female flies are easily distinguishable. Also, it has many types of hereditary variations that can be seen with low power microscope. Then linkage. Here, Morgan attributes this due to the physical association of the linkage of the two genes and coined the term linkage. And to describe this, physical association of genes on a chromosome or termed as recombination, we describe it in a proper way. Morgan and his group also found out that even when genes were grouped on the same chromosome, Some genes are very tightly linked, which shows a very low recombination, while others will loosely linked, they show high recombination. And percentage we can find out 1.3% and 37.2% recombination are found out. Students, Alfred Schwarzband used the frequency of recombination between the gene pair on the same chromosome as a measure of the distance between the genes mapped their position on the chromosomes. And nowadays genetic maps are used as in the Human Genome Project, which will be described in previous next chapters. Here again cross A, as you can see, and with these crosses you can find out here in cross A chromosomes are in near, and here in cross B are far away, and with this though they are near, recombinant type will be very less, and the far away recombinant type will be very high. Polygenic inheritance. Before starting this, let's see what the PDF says. Sorry, we already discussed about this polygenic inheritance, and here you can see Mendel's studies mainly describe those traits that have distinct alternate forms such as flower color, which are either purple or white. But if we look around, then there are also many different type of color flower are there. And if you come into human beings, there are not only just tall or short people, but there are also many medium height peoples are there, and such traits are generally controlled by 3 or more genes and thus called polygenic traits. Besides the involvement of multiple genes, polygenic inheritance also takes in account the influence of environment. Human skin color is the proper example of this. And in a polygenic trait, the phenotype reflects the contribution of each allele, that the effect of each allele is additive. So let us assume same example is taken, 3 genes ABC, then dominant allele ABC will be the darkest skin color, while the receive allele will have the lightest skin color, and the mixed one will have the intermediate skin color. Next, pleiotropy. Pleiotropy means a single gene can exhibit multiple phenotypic expressions. So genes are called pleiotropic genes, and there are some examples. The disease is called phenylketonuria, PKU. In shortcut is called PKU, which occur in humans. This disease is caused by mutation in the gene that codes for the enzyme phenylalanine hydroxylase, single gene mutation. This manifests itself through the phenotypic expression characterized by mental retardation and reduction in the hair and skin pigmentation. So, it is caused by mutation in the gene that codes for enzyme phenylalanine hydroxylase. Remember, single gene mutation, and it causes mental retardation as well as reduce in hair and skin pigmentation. Next is sex determination. Let's see the PDF, what it says here. Autosomes and sex chromosomes or allosomes. The chromosomes that are involved in sex determination are called sex chromosomes or allosomes. They include X and Y chromosomes. Autosomes are chromosomes other than sex chromosomes. Number of autosomes is same in male and female. As you can see, 1 to 22 are autosomes, and the only one pair it is the sex chromosome, X and Y mentioned. Here, Henking 1891 studied spermatogenesis in some insect. Observed the 50% sperm receive a nuclear structure after spermatogenesis, and other 50% sperm did not receive it. Henking called this structure as X body. Now it is called X chromosome. Mechanism of determination: XX and XO mechanism found in insect, XX XY mechanism found in also type of insect, ZZ ZL mechanism found in birds. First of all, XS and XO mechanism. Here, the male is heterogametic, that is X and O, gametes with X and gametes without X. And female is homogametic, XX. All gametes are with having X chromosome. Example, many insects such as grasshopper. Here you can see 22 plus X and O, 22 plus X and X. Next is XX and XY mechanism. Male is heterogametic, X and Y, female is homogametic, only X. Example, human and Drosophila. See the diagrams. Next is Z and JW mechanism. Male is homogametic, ZZ, and female is heterogametic, Z and W. Example, birds. XS and XO and XS and XY mechanism shows male heterogamety, while the ZZ and ZW mechanism shows the female heterogamety. Coming to human being, we already doing this, but again it is given, as you can see. Humans has 23 pair of chromosomes, where the 22 pair are autosomes and 1 pair will be the sex chromosome. A pair of X chromosome is present in female, while X and Y chromosomes are present in male. During spermatogenesis, male produces two types of gametes, 50% to the X and chromosome and 50% to the Y chromosome. Female produce only ovum with an X chromosome. And then there is an equal probability of fertilization of ovum with the sperm carrying either X or Y chromosome. The sperm determines whether the offspring will be male or female. Coming to honeybee, here what happens? It is based on the number of set of chromosomes of an individual receives. Here queen will be diploid, drone will be haploid, and here you can see drone will be haploid like this and worker bee will be diploid like this. Parents, because female having 32 chromosomes, male having only 16 chromosomes. In female body meiosis occur, 16 occurs there. In here mitosis occur, 16 chromosomes are there. F1 generation, male having 16 chromosomes, female having diploid or 32 chromosomes. Fertilized egg develop as female, queen or worker bee, and unfertilized egg develop as a male, that is drone. It is called parthenogenesis. Therefore, the female are diploid having 32 chromosomes and male are haploid having 16 chromosomes. This is called haplodiploid sex determination system. In this system, the male produces sperm by mitosis. They do not have father, thus they cannot have son, but they have grandfather and can have grandsons. Mutation. Before starting this, let's see what the PDF says, NCERT says. Sex determination, same points are given. Name is Hanking 1891, and who give the term X body? Remember, question asked you. And large number of insect of mechanism shows the sex determination X and O type. That is, all egg bear an additional X chromosome beside the other chromosomes. On the other hand, some of the sperm bear the X chromosome, whereas the some do not. Eggs fertilized by sperm having X chromosome becomes female. Those fertilized by sperm which do not have X chromosome, they becomes male. Same with the in grasshopper, XO type of sex in which the male have only one X chromosome beside the autosomes, whereas female having pair of X chromosomes. And coming to mammals, including men, XY and XS type, we already seen. And in birds there is different mechanism shows where the female shows heterogamety, male shows homogamety. ZZ and JW formation. And here again the human beings, 22 are autosomes, one pair will be sex chromosome, and there are 50% chance that the sperm carrying X chromosome or 50% chance the sperm carrying Y chromosome. If the X chromosome fuses with the female ovum, then it will be a girl child, and if the Y chromosome carrying sperm fuses with the ovum, it will be the male child. Next, in honeybee, you can see an offspring found from the union of a sperm and an egg develop as a female, either queen or worker, and an unfertilized egg develop into a male that is drawn by means of parthenogenesis. This means that male having only half number of chromosomes that of the female. The female are diploid having 32 chromosomes and male are haploid having 16 chromosomes, as you can see in this diagram. And that's why the male, they do not have father, thus they cannot have son, but they have grandfather and can have the grandsons. Mutation is there. Let's go to the PDF. Mutation. It is a sudden heritable change occurring in the DNA sequences that result changes in the genotype and the phenotype of the organism. Sudden changes occurring during the DNA sequences. Recombination and mutation leads to variation of DNA. There are types of mutation: point and frame shift mutation. First of all, point mutation. Here, in this mutation due to change in single base pair of DNA, example sickle cell anemia. It's the original sequence, as you can see, and in point mutation, it is different. Coming to frame shift mutation, it is a deletion or insertion of base pair resulting in the shifting of DNA sequences. Loss or gain of DNA segment causes chromosomal abnormalities, and chromosomal aberrations are seen in cancer cells. Like this you can see, point mutation. This topic we will cover in the next chapter with the proper examples. Mutagens. Mutagens are the agents that induce mutation, and types are two types: physical, chemical mutagens. Physicals are UV radiation, alpha, beta, gamma rays, X-rays, etc. And chemical mutagens: mustard gas, phenol, formalin, acetic acid, formic acid, and NH3, etc. Then is pedigree analysis. Here, in human, controlled crosses are not possible, so that the study of family history about inheritance is used. So the analysis of genetic trait in several generations of a family is called pedigree analysis. Here you can see family tree. The representation or chart showing a family history is called family tree or pedigree. In human genetics, pedigree study is utilized to trace the inheritance of a specific trait, abnormalities or diseases. First of all, remember the symbols. Male having this type of square symbol, female round symbol, sex unspecified, affected individual, mating, single line between there, mating between relatives, double line is there, parent above the children below, parent, children, parent with affected male child, parents, normally a male child affected, 5 unaffected offspring, this symbol. Here, pedigree analysis helps to understand whether a trait is dominant or recessive. Pedigree analysis of autosomal dominant trait. Example, myotonic dystrophy. Myotonic dystrophy, as you can see. Here, mother is affected, father unaffected, and the children are like this. Offsprings are like this, you can see. Affected. Then, this generation not affected, this will be affected. Myotonic dystrophy. Again, autosomal trait is sickle cell anemia. All the RBC will be like this, sickle celled. Both are normal. Either one is a carrier, and here affected persons like this. They all have sickle cell anemia. Queen Victoria was a carrier of haemophilia, so her family pedigree shows many hemophilic descendancies. As you can see, carrier, for carrier, the symbol is taken Queen Victoria, Prince Albert, and here it is a carrier, affected hemophilic male, again carrier female, and in George V cases, here again carrier female, affected male, carrier female, carrier female, carrier female, affected male. Here also affected male, affected male, affected male, affected male, affected male, like this. Genetic disorders. Before starting this, go to the NCERT, what it says about mutation. Mutation is the phenomenon which results in alternation of DNA sequences and consequently results in changes in the genotype and phenotype of an organism. And here in mutation, you all know that the DNA helix runs continuously from one end to the other in each chromatid and is highly supercoiled form. Therefore, loss or gain of a segment of DNA result in the alternation of chromosomes. And this is called abnormality or aberration. Chromosomal aberration are commonly observed in the cancer cells. And in addition to this above, mutations also arise due to change in a single base pair of DNA. It is called point mutation. Example, sickle cell anemia. And deletion of insertion base pair of DNA is called frame shift mutation. We will see in the chapter 5, next chapter. And coming to mutagens, UV radiations causes mutations in organism. It is a mutagen. Pedigree analysis. Here you can see symbols are there. Pedigree means a family tree. Okay. So to analyze a trait in a several generation of a family is called pedigree analysis. Like this. Alternation of or change in genetic material is referred as mutation. Again, mutation is there, and nothing more. On to disorders. Here, broadly, genetic disorders may be grouped into two categories: Mendelian disorder and chromosomal disorder. Coming to Mendelian disorder are mainly determined by alternation or mutation in a single gene. Remember, Mendelian disorder are mainly determined by alternation or mutation of a single gene. These are transmitted to the offspring on the same line as we have studied in the principles of inheritance. The pattern of inheritance of such Mendelian disorder can be traced in a family tree by pedigree analysis. Most common and prevalent Mendelian disorder are haemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia. And it is also mentioned that the Mendelian disorders are dominant or recessive. Only by pedigree analysis we can easily understand either it will be dominant or recessive. Similarly, the trait may also be linked to the sex chromosome, as in case of hemophilia. Evidence that the X-linked recessive trait shows transmission from carrier female to the male progeny. And coming to color blindness, it is a sex-linked recessive disorder due to the defect of either red or green cone of eye, resulting in the failure of discriminate between the red and green color. This defect is due to mutation in a certain gene present in the X chromosome. It occurs in about 80% of male and only about 0.4% of females. Why? Because genes that leads to red-green color blindness are on the X chromosome, and male have only one X chromosome and female have two. The son of woman who carries the gene has a 50% of chance being color blind. The mother is not herself color blind because the gene is recessive. This means that it affects the suppressed by her matching dominant normal gene. A daughter will not normally be color blind unless her mother is a carrier or her father is a color blind. And coming to hemophilia, the sex-linked recessive disease which shows a transmission for unaffected carrier female to some of the male progeny. In this disease, a single protein, that is a part of the cascade of proteins, cascade means group of proteins involved in the clotting of blood, is affected. And due to this, in an affected individual, a simple cut will be result in non-stop bleeding. The heterozygous female will be carrier for haemophilia, may transmit the disease to the sons. And this is why, because the hemophilic in extremely rare, because mother is such a female, has to be at least carrier, father should be hemophilic. And the family pedigree Queen Victoria, so the number of hemophilic descendant as she was the carrier of this disease. Come to sickle cell anemia, this is an autosome linked recessive trait that can be transmitted from parents to offspring when both the partners are carrier for the gene. And the disease controlled by single pair of allele HB and HBS. Out of the three possible genotype, only homozygous individual HBS. So the disease phenotype. Heterozygous individual appear apparently unaffected, but they are carrier of disease, and there is 50% of probability of transmission of this mutant gene to the progeny. Thus, exhibiting sickle cell trait, the defect is caused by substitution of glutamic acid by valine at the 6th position of the beta globin chain of the hemoglobin molecules. See the diagram? It is the normal hemoglobin and sickle cell hemoglobin, where in the 6th position here glutamic acid converted by valine. Because of this particular single one, the RBCs become different and sickle celled. Then same points are given. Coming to phenylketonuria, the inborn error of metabolism is also indicated as autosomal recessive trait, and lack of enzyme that convert the amino acid phenylalanine to tyrosine. As a result, the phenylalanine is accumulated and converted into phenylpyruvic acid and the other derivatives, and accumulation of this in the brain result in the mental retardation. They are also excreted through the urine because of its poor absorption by the kidney. It is the phenylketonuria. Coming to thalassemia, this is also an autosomal linked recessive blood disease transmitted from parent to the offspring when both the partners are unaffected carrier for the gene. This defect could be due to either mutation or deletion, which ultimately result in the reduced rate of synthesis of one of the globin chain, alpha or beta chain, that make up the hemoglobin. This causes anemia, and thalassemia can be classified according to which the chain of hemoglobin molecules are affected. In alpha thalassemia, production of alpha globin chain is affected, while the beta thalassemia production of beta globin chain is affected. Alpha thalassemia is controlled by two closely linked genes HBA1 and HBA2 on chromosome 16 of each parent. It is observed due to the mutation or deletion of one or more of the four genes. The more genes are affected, the less alpha globin molecules are produced. While the beta thalassemia, it is controlled by single gene HBB on the chromosome 11 on its parent and occurs due to the mutation of one or both the genes. There are some more disorders. Let's see. First of all, genetic disorders. Genetic disorders are the disorders due to the change in the genes or chromosomes. Here, Mendelian disorder, haemophilia, sickle cell anemia, color blindness, phenylketonuria, thalassemia, cystic fibrosis. Chromosomal disorder, Down syndrome, Klinefelter syndrome, Turner syndrome. Let's see. First of all, genetic disorder. This is only caused by alternation or mutation of only single gene. Haemophilia, color blindness, sickle cell anemia, phenylketonuria, thalassemia, cystic fibrosis. First of all, same diagrams are given. Here, haemophilia or royal disease, sex-linked disease, X-linked recessive disease in this protein is involved in the blood clotting is affected. A simple cut result to the non-stop bleeding. It is controlled by pair of allele, capital N and small h. Capital H is normal allele while the small is the responsible for haemophilia. Normal female, heterozygous female, homophilic female, normal male, hemophilic male. Why it is called royal disease? Because it occurs during the Queen Victoria, and we already discussed about this. Color blindness. It is a sex-linked disease due to the defect in either red or green cone of the eye present in the retina. It results in the failure to discriminate between the red and green color. It is due to the mutation in some genes in X chromosome. It occurs 8% in male, only 0.4% in females. Due to the gene is X-linked. Here you can see normal color vision. It can easily identify the colors, and color blindness, everything he found out the green only. Here in color blindness you can see carrier women, normal men, and seeing the crosses you can see normal female, carrier, normal, and color blind is there. Here carrier women, color blind men. Here what happened? Carrier female, color blind female, normal, color blind. That's why normal allele is dominant. Recessive allele causes color blindness. The son of heterozygous woman will be carrier and has a 50% chance being color blind. The daughter will be color blind only when her mother is at least carrier and her father is color blind. Next, sickle cell anemia. Here you can see it is autosome linked recessive trait. It can be transmitted from parents to offspring when both the parent partners are carrier. This disease is controlled by a pair of allele HB and HBS. Homozygous dominant, normal, heterozygous carrier, sickle cell trait, homozygous recessive affected. See the diagram. And this defect caused by substitution of glutamic acid GLU by valine at the 6th position of the beta globin chain of hemoglobin. It is the beta globin chain, and where what happened? The glutamine is substituted by valine, and because of this they have sickle cell anemia. And here it is a normal RBC, it is a sickle cell RBC, abnormal RBC, and they causes blockage like this. The mutant hemoglobin molecule undergoes polymerization under low oxygen tension, causes the change in shape of the RBC from biconcave disc to elongated sickle cell structure. Biconcave disc to elongated sickle cell structure. Coming to PKU, phenylketonuria, it is an inborn error of metabolism, autosomal recessive trait. It is due to the mutation in the gene that codes for the enzyme phenylalanine hydroxylase. This enzyme converts the amino acid phenylalanine into tyrosine. Here you can see a normal person, dietary protein phenylalanine hydroxylase, tyrosine, melanin protein dopamine is produced. But in PKU patient, it directly converts into phenylalanine and then phenylpyruvic acid. Phenylpyruvic acid. Okay, and they have PKU. The affected individual lacks the enzyme. As a result, phenylalanine accumulates and to convert into phenylpyruvic acid and other derivatives. And what is the result? Phenylpyruvic acid and other derivatives accumulate in brain, resulting in mental retardation. This is also excreted through urine because of poor absorption by the kidney. Coming to thalassemia, it is an autosomal linked recessive blood disease. It is transmitted from unaffected carrier parent to the offspring. It is due to the mutation or deletion. It results in reduced synthesis of one of alpha or beta globin chain of hemoglobin. It forms abnormal hemoglobin and caused anemia. Thalassemia is a quantitative problem, synthesis of few globin molecules. Sickle cell anemia is a qualitative problem, synthesis of an incorrectly functioning globin. There is a difference between thalassemia and sickle cell anemia. In thalassemia, based on the chain affected, here you can see it is autosomal linked recessive disease and transmitted from unaffected carrier parents to the offspring due to mutation or deletion. Result the reduced synthesis of one of the alpha or beta globin chain in hemoglobin. It forms abnormal hemoglobin, causes anemia. Here is the difference between thalassemia and sickle cell anemia. Thalassemia is a quantitative problem. These are few globin molecules, and sickle cell anemia is the qualitative problem, synthesis of an incorrectly functioning globin chain. First of all, thalassemia. Here the production of alpha globin chain affected. It controls by two closely linked genetic genes HBA1 and HBA2 on chromosome 16 of each parent. HBA1 and HBA2, chromosome 16 of each parent. Here you can see RBC, alpha chain, beta chain. Mutation or deletion of one or more the four genes causes the disease, and the more genes affected, the less alpha globin molecules are produced. Coming to the beta thalassemia, here production of beta globin chain is affected, and it is controlled by single gene HPP on chromosome 11 of each parent. Mutation of one of both the genes causes the disease. Chromosomal disorder. Here, the disorder caused due to the absence or excess or abnormal arrangement of one or more chromosomes. Chromosomal disorder, aneuploidy, polyploidy. These are two types. First of all, aneuploidy. In aneuploidy, it is the gain or loss of chromosomes due to failure of segregation of chromatids during cell division. Aneuploidy. See this, you can see meiosis 1 occur in both the sides. Here what happened in second meiosis, all having the haploid genetic material, but here due to the failure of segregation of chromatids, what happened? Here one of chromosomes are more and one is less. It is called aneuploidy. It is a 4 type: nullisomy 2n-2. A chromosome pair is lost from diploid set. Monosomy 2n-1. A chromosome is lost from diploid set. Monosomy, like this you can see. Trisomy 2n+1. A chromosome is added to the diploid set. Trisomy. Tetrasomy 2n+2. 2 chromosomes are added in the diploid set. Tetrasomy. Polyploidy or euploidy. It means the increase in a whole set of chromosomes due to failure of cytokinesis after telophase stage of the cell division. This is very rare in human but often seen in plants. Polyploidy or euploidy. Here the parents, you can see diploid cell, meiotic error occur, unreduced gametes, cell fertilization, zygote formed, 4 n chromosomes are tetraploid is formed. Mostly it is very rare in human but often seen in plants. Next, Down syndrome or Mendelism. The presence of an additional copy of chromosome number 21, trisomy of 21. Genetic constitution 45A autosomes plus XX or 45A plus X and Y, that is 47 chromosomes. Here you can see trisomy of 21th chromosome. It occurs Down syndrome or Mendelism. Features are short stature, small round head, broad flat face, furrowed big tongue and partially open mouth, like this. Broad palm with characteristic palm, simian crease, many loop on fingertip, retarded physical, psychomotor and mental development, congenital heart disease. These are the features. Here again see the diagrams like this: simian crease, normal palm crease, Down syndrome, and here again flat facial face and protruding tongue, flat nasal bridge, simian crease, gap between first and second toes. Klinefelter syndrome. Klinefelter syndrome. It is the presence of an additional copy of X chromosome in male. Additional copy, that is 44 autosomes plus XXY, that is 47 chromosomes. It is a normal male and it is a abnormal male, or it is sterile, like this in the diagram. This body height, breast enlargement, here you can see. Features: overall masculine development, however feminine development, that is the development of breast, also called gynecomastia, is also expressed, and sterile and mentally retarded. Next is Turner syndrome. This is the absence of one of the X chromosome in female. Coming to genetic constitution, 44 autosomes and X and O, that is 45 chromosomes. Normal one, abnormal one, and they have saw-started webbed neck, sail chest, underdeveloped breast and widely spaced nipples, rudimentary ovaries, brown navy. Features are sterile, ovary rudimentary, lack of other secondary sexual characters, dwarf, mentally retarded. With this, we have completed this chapter from this PDF. Now, last time, go to the NCERT. Here, aneuploidy is given, polyploidy is given, same points, listen, nothing more. Diagrams: trisomy or monosomy of chromosomes, Down syndrome, Turner syndrome, Klinefelter syndrome are example of chromosomal disorder. Coming to Down syndrome, and additional copy chromosome number 21, that is trisomy of 21, causes Down syndrome. This was first described by Langdon Down in 1866. Remember, and features are short-started, small round head, furrowed tongue, partial open mouth, palm broad, palm crease. Physical, psychomotor and mental development is retarded. Coming to Klinefelter syndrome, this is caused due to presence of an additional copy of X chromosome resulting in the karyotype, 47 chromosomes, X, X and Y. Such individuals have overcome masculine development, however feminine development also shows development of breast, that is gynecomastia, also caused because of Klinefelter syndrome, and such individuals are sterile. Turner syndrome, such a disorder caused due to absence of one of the X chromosome, 45 with X and O. Such females are sterile as ovaries are rudimentary or beside other features including lack of other secondary sexual characters. It is Turner syndrome. And with this, we have completed this chapter. As I told you, it is a long one and many important questions are coming from this chapter. So, go through this, both the PDF. I hope the PDF will also helpful, as well as the NCERT line by line topics are we have completed, and this chapter is completed. With this, the next chapter we will give some more new diagrams in the chromosomal theory of inheritance, and we will cover another important topic in the next video. So till then, thank you, and new viewers, do like this channel for further videos, and do not forget to press the bell icon for new videos uploaded, and comment if you want. If you find any doubt, simply ask. My number is also given in the description box. And new viewers, please subscribe my channel. Thank you. Jai Jagannath.